Hello, everyone, to this new edition for the first Inria Talk of 2025. We have Clemence here, and thank you for accepting such hurry. Clemence is a junior researcher at Inria in Bordeaux, right? And she's going to be presented today to us an exploration of microbial ecosystem from compositional patterns to metabolic models. We have a lot of work in this, like a common ground on this, so we are looking forward to your presentation. Thank you very much, and I let you present. Thank you.
Good morning, everyone. Thank you, Luis, for the introduction, and thank you to all in Inria Chile for the organization and the invitation. So I will present you computational models that we develop in the team to study microbial ecosystems. So microbial ecosystems, they are absolutely everywhere, from the inner of our gut, in every animal, on our skin, in the ocean, associated to plants, in the soil. So they are everywhere. They are sometimes referred to as microbiota or microbiomes. And they are very complicated to understand. They can be understood or studied through the paradigm of systems biology, a conceptual framework that motivates the analysis of these living organisms, the cells, through the integration of the study of each individual components and the study of them as a whole that has interactions among components. Systems biology promotes the analysis of these organisms through a virtuous cycle that associates three concepts: the experimentation, the generation of data at a high throughput, and the use of computational models. So experimentation will generate the data that we call omic data. It's heterogeneous, it's large scale. For instance, you can study the DNA sequences, that's the genomics data, the expressed genes, that's transcriptomics, the molecules, metabolomics, and all of this data is being integrated into dedicated computational models in order to generate predictions, hypotheses on the mechanisms of the living organisms, and to guide further experimentation, hence the virtual cycle. In the team, we use several types of models depending on the questions we want to address and depending on the challenges we have to face. So among the challenges, there is the question of complexity reduction, scalability to large systems, need for accuracy, difficulty of data integration. And among the models that we use, there is statistical learning, numerical models, or knowledge representation and reasoning that I will all three present today. First, for a minute, let's forget about bacteria and microorganisms and look at these pictures. We as humans are really good when it comes to classify, recognize, distinguish things. So if I show you these pictures, you will probably very easily classify them and tag them, label them, for instance, as forest for two of them, even if they are coming from different seasons, and as grasslands for the two others. So those would be the ecosystem types that we use to label the pictures. And we recognize these ecosystem types because of the species, the animals, the vegetations that we see in the picture. So the tree for the forest, some animals from the grassland. Each of these ecosystems harbors specific species. So for large-scale ecosystems, it's quite easy to classify. For microbial ecosystems, we don't have these pictures. Hence, it's more complicated. The pictures that we have, they come from genomic data, mostly, that enables us to get information about who is here, what are the bacterial species, the microbial populations that are present. And from this, works from the early 2010 used clustering to define the ecosystem types of the gut microbiome, which were defined as enterotypes. So 3 enterotypes were identified, each dominated by a given taxonomic group, Bacteroides, Protella, or Phoenicutes. And they became an integral part of gut ecology in order to understand what a normal, a healthy gut microbiome is, and to identify deviation from health. But in fact, it's not that easy because it's hard clustering that enterotypes uses. And the number of clusters, it's not always easy to decide. And there are also discussions on whether it's actually a gradient that is important to define the phenotype. What hard clustering doesn't address is the continuum between those assignments. For instance, if we go back to the grassland and the forest, there are some pictures that we might struggle to classify, to label with our eyes. And it's very important yet to classify them because many pictures might actually be in the continuum. So what we aimed is to build a model that addresses this continuum and is able to quantify the different clusters in each of the pictures. So that is what we did for the gut microbiome in order to define signatures instead of enterotypes that would take into account the gradients. So our picture that we work with is the data that you have on the bottom left. It's the relative abundance of taxonomic groups in the samples. So the columns will be the samples. We had more than 5,000 here. And the rows would be the taxonomic group. Here we are at the genus taxonomic rank. So if you analyze this data, you can just simply visualize these pictures. It's the relative abundance that you have on the right bottom. But we have many features, up to 500. So it's very complicated to distinguish the role of each feature in the composition. So you can go higher in the taxonomic rank, which will reduce the number of features at the top right here. But then you lose some precision. So what hard clustering did in enterotype is assigning a cluster from the genus level composition. And what we did in the project is to present gradients, profiles of assemblage that we call enterosignatures that we build directly from the data at the genus level. So we use non-negative matrix factorization in order to generate those enterosignatures. NMF will provide you with two matrices, one that describes the composition of the profiles, the enterosignatures, using an assembly of the original 500 features. And the second matrix will give you the relative abundance of each enterosignature in the sample. So this is the composition of the 5 enterosignatures that we identified. So you have them on the left, dominated by a specific taxonomic group, and an enterosignature is then a microbial guild, an assembly of microbes that tend to co-occur in samples. And then a sample will be described as a weighted assembly of these 5 enterosignatures. But it can be not necessarily the 5 enterosignatures, it could be 1, 2, or 3, or 4. So we looked at our original data. That's all the points that you see in the PCOA here. And we assessed whether, how many signatures were needed in order to properly describe the samples. So here you have 1 to 4. And if hard clustering is enough, most samples will be described by a unique enterosignature. And you see that it's not the case. Actually, for more than 70% of our data, 2 or 3 enterosignatures are needed to properly describe the sample. So our soft clustering approach is actually useful to describe with precision data. Associated to data, we have metadata describing the phenotype of the host, whether the host individual took medication, what diet was associated to the individual, the age, the BMI. We have many metadata that we can associate to the enterosignatures. And we could find correlations between some healthy phenotypes and the prevalence of the firmicutes enterosignature, or some phenotypes associated to perturbed microbiome composition and some other enterosignatures. But then the question arises of whether these signatures can be reused for new data. So we tested a new cohort, and we either reconstructed de novo the same intro signatures, so we learned again, or we reused the signatures that we used in the first learning cohort. And you can see in the middle bar plot that actually reusing the previous signatures gives good model fit scores to describe the new samples. So it means that we don't need to learn again and that we learned generalizable signatures in the first place. So having this in mind, we wanted to look what happens when we study cohorts that are quite different from the learning cohorts. It's well known that Western populations and non-Western populations have varying microbiome composition. And we tested whether we could capture this using our model. So on the left part of the plot, you have the learning cohorts that we used in green. And on the right part, the non-Western populations that we studied with the same signatures that were learned on the left data. And you can see that for most of the data, the model fit quality score is pretty good, except for 3 cohorts at the end, which was a bit surprising. So we went back to the papers, and we actually saw that these 3 cohorts were not preserved in ice after sampling, but in ethanol, which could explain the varying composition of microbial populations and the fact that the model doesn't fit. But for the rest of the cohorts, it fits pretty well, and we retrieve some features that we are expecting in these populations. For instance, in the adults composition in the bottom, you see a prevalence of the orange signature, that's Prevotella. It's really prevalent in these populations, whereas in Western populations, on the left part, you have a high abundance of Bacteroides and Firmicutes entero signatures. But then there are still some samples that are not very well described by the model. And why is that? We studied the metadata we had for these samples in order to understand better. And we noticed that these ES entero signature atypical compositions were associated to perturbed gut microbiome composition. So the model fitting score decreases in preterm infants compared to infants born after 9 months. It decreases in C-section delivery birth mode. It decreases after antibiotic treatment. So actually, the fact that the model fits more or less can be used to identify and to describe perturbations of the gut microbiome composition. So overall, with this project, using statistical learning, we described a unified model of the gut microbiome composition. And the good thing about it is that it can be applied to novel cohorts, just as it is, no need to learn again. It can associate to host phenotypes. It can bring some information about perturbed microbial composition. And we studied a bit in the paper the functions that were carried by each of the entero signatures, whether they had specific functional niches. I won't go into the details here, but I will use this as a transition because it's important to know not only who is present in terms of bacterial populations in a microbiome, but also what they do, what functions can they do in the ecosystem. And that is actually the core of my research. And I will focus on this now. So just to go back to systems biology for a bit, you have a bacterium here. It can be studied at several levels, the genes, the expressed genes, the metabolites, and all of this has to be combined together in order to properly understand what's going on in the bacterium and taking into account the environment. But here we're not just looking at a unique bacterial population, we are looking at bacterial communities. So it makes the system even more complex, and you have to take into account the interactions among the members of the community. And actually, most of the interactions are mediated by metabolism, that is the collection of functions, metabolic reactions, transforming molecules into others in the cells. So by looking at the metabolism, maybe we can get some hints about what occurs within the communities. So that's what we do with metabolic modeling. That start with the genomic sequences, the DNA of any organism, from which we identify genes, proteins. We tag functions on these genes, and we use them to build what we call the metabolic network. So that is a metabolic network. Usually at the genome scales, it has a few thousands of metabolic reactions. A metabolic reactions would be in the form of what you see in the figure here. A plus 2B would give 2 molecules of C plus 1 molecule of T. And in the figure on the right, you can see reactions in blue triangles, metabolites in yellow circles. You see that it's already hard to visualize, and here you have only 90 reactions. So we need models more than visualization in order to properly understand metabolism. And these models, they will rely on the metabolic network on one side, and they will account for additional data, for example, describing the environment. And all of this will be integrated into a mathematical or computational model in order to generate predictions, such as the growth of the bacteria, interactions among the community, or predictions of medium. So what model to use is always an important question. And actually, in the past 10 or 20 years, there's been a rise in non-optimal use cases for modeling. Sequencing gets cheaper, which is great. We can sequence many species, many bacterial communities, get what we call metagenomic data. But for much of these communities, much of the species, the populations, we know very little. Sometimes we know nothing. The only information that we have is the genetic sequences that we get. There is no biological expertise, no scientific literature, so it's really complicated to validate. And then we are also working at larger scales than what we used to do. So manually curating, enhancing the models, the network is getting complicated when you have hundreds of networks to work with. There are some functions that are entirely missing, or for which there is uncertainty. And then there is the matter of scalability. So choosing the mathematical or computational model for the simulations is really important, depending on these constraints and on the biological questions that we address. So in the team, what we do is reconstruct metabolic network for all members of a community, and then we choose the model depending on the question and on the constraints of the community. So if we work with small communities on which we can make some manual curation, taking into account literature, expertise, we use constraint-based modeling. So we build numerical models associating numerical values to each of the metabolic reactions in order to quantitatively predict the activity of the reactions. And if we work with large communities for which we just work with automatically generated data, we cannot perform curation, then we use simpler models just to reduce the complexity of the data. And we use reasoning approaches. So no longer numerical models, but rather Boolean abstractions, 0 or 1, to describe the activity of functions. So I will start with the numerical models and present you briefly a few projects. And first, how we can make dynamic models of small controlled bacterial communities. So such dynamic models would be following this framework. So you have the metabolic network on the left. You use this numerical model that I introduced earlier. So constraint-based modeling, such as flex balance analysis. You add the environmental condition description. You add constraints, thermodynamical constraint. You add the assumption that you are at steady state. And you optimize the flux value, the activity, if in one of the reactions of your model. And if you do this at each time step, and you integrate over time the results in an ordinary differential equation model, you can get what we call dynamic flux balance analysis models, in which you can follow over time the concentrations of molecules, the populations of each of the microbial members. That is what we did during the PhD of Maxime Lecomte. It's very cliche, but for French people, but we studied cheese. So starting from a community of 3 bacteria, we studied the production of aroma molecules in the cheese over a 7-week process. So we started with the description of milk as the environment. We had a metabolic network for each members. We had data, the genomes, metabolomic, transcriptomics, all this omics data. And we wanted to understand the mechanisms responsible for the production of aroma compounds. So our strategy was to build individual models for each of the species, calibrate them, like training step on pure cultures, and then testing would be the prediction of community dynamics. So we used a learning-based strategy. So Maxime did a lot of curation on each of the models in order to highlight the metabolism of the species that was specific to the milk condition. And then he built the first dynamic models for the individual community, the individual species. So you can see in green line the model, in orange the original data. So for individual growth, the results are quite precise. The question is whether from these models we can accurately describe the community. And the answer is overall yes. On the left, you have the results of the training dataset on individual cultures. And on the right, you have the community model. We reused the models that we trained on pure cultures, and we tried to predict the data that we had in the community. So except for a few predictions, overall, we predicted very well the populations of bacteria and the concentrations of metabolites. And from this, we can get precise mechanisms on what could occur in the community during cheese production. So we have the concentrations of molecules over time on the top graphs. And for each molecule, we know which member of the bacterium, of the bacterial community, is responsible for the production or the consumption. So that's a mechanistic model that's really useful to raise predictions. And it integrated a lot of data, but it was quite complex. It took a lot of time to get built. So it's not scalable for large communities. The second project that I wanted to introduce on this topic is related to statistical learning on this data. Because, as I told you earlier, flux balance analysis solves an optimization problem. And this optimization problem has to be done at each time point, which can be very time consuming for large systems. So our idea in this project, led by the postdoc Pablo Galdetes Salas, an engineer Coralie Müller, and a fellow researcher Simon Labarthe, was to accelerate the process of building dynamical model by skipping this optimization step and rather use statistical learning. So our application here was no longer the cheese, but the infection of a pathogen, Salmonella typhimurium, in the gut microbiome, in the human gut microbiome. That is a process that is very well known by biological experts. We know how the microbiome works when there is no infection. We know the perturbations that the bacterium is likely to trigger when it arrives and how it can alter the system. So we translated what we know into metabolic networks and tracked metabolic pathways that were associated to the infection. So here you have 3 systems. The purple one is the commensal, so the nice bacteria of the gut microbiome. We have the human host on the bottom, so the cells of the intestine. And we have in red the pathogen. And the idea was to build a surrogate flux balance analysis model that does not need optimization at each time step. So we would rather learn a database a priori in which from environmental conditions, we can predict the flux activities, the value of dedicated metabolic reactions in which we are interested in. And then during the computation, we don't have optimizations no more, and we can directly get the output fluxes value. And it works pretty well. So here you have several plots showing the concentrations of several molecules, the populations of the three partners. And in lines, you have the simulations resulting from the optimizations, the dynamic FBA, the classical one. And in dotted lines, you have the predictions of our model. So overall, our model that is statistical learning works very well. We have less than 2% error. But the good thing is that we have a 60 times speed up. And that is really useful because what we want to do is study this mechanism at a spatial scale. So we would need even more optimization to be solved because we use not only time, but also space. And that is the work that is ongoing. So all of these models that I presented so far, they work with small communities. So it's not really scalable. And when it comes to larger communities, we use an entirely different type of modeling that is reasoning based. So first, I will present this topic and a first project. So the model is actually quite simple. On the left, you have a metabolic network with the cell that describes the organism. You have a few reactions that are the squares, and these reactions consume metabolites in circle and produce others. You have two metabolites outside the cell, L and B, which are what we call the seeds. They describe the environment. And from this, we detect what is reachable in the metabolic network. So we identify the subnetwork associated to these seeds. If we change the seeds, we change the subnetwork that is reached, that is activated. So it's very simple. It's just a matter of reachability in graphs. And it's said to be, early papers show that it's quite accurate when compared to numerical models, and that it's resilient to incomplete data, which we have when we work with automatically generated metabolic networks. We work on this using symbolic artificial intelligence that is used to make this modeling. So we use answer set programming. That's from the field of knowledge representation and reasoning. It's a logic paradigm in which you describe the problem and you leave the solving to the solver. So you really don't mind about how it's going to be solved. All you have to do is properly design your problem, your available information as sets of rules and constraints. So as an example, how can we describe what is rich in a metabolic network from the description of the environment, the seeds? So the first part of the code here describes the reactions. A reaction has a name. Reactions have input, reactants. For instance, A is the input to reaction R1. It has products. B is the output of reaction R1. We have the seeds A and C. And we have just two rules to describe the reachability of compounds from the seeds. So it's really efficient. That's not machine learning anymore. That is machine reasoning, and it also supports optimization. So we work a lot with this paradigm. And Shamnam Ghasemi-Nejad, a PhD in the team, tries to solve the opposite problem, the inverse problem of what I explained earlier. That is, usually you predict the reachable compounds from the description of the environment, and Shamnam tries to predict the environment from a description of what occurs in the cell, or what is supposed to occur in the cell. So she designed and developed a tool called Seed2LP that uses reasoning in order to predict these sets of seeds. But additionally, because numerical models are very widespread and used by the community, she designed hybrid solving methods that would filter or generate solutions that are not only compatible with network expansion, the discrete models, but also with the numerical paradigm that I presented earlier. So in the paper, we compared the tool of Shamnam here in hybrid mode, so ensuring network expansion, reachability, and steady-state satisfiability for FBA, to a MILP implementation that is available in a tool. And the first observation was that actually reasoning really helps to get solutions fast. Getting one solution we can do very fast, but the MILP implementation also can do it before the timeout. And actually, our tool times out for a few networks. But then if you want more than one solution, which is likely to be the case when you work with biologists, there is no reason that the first solution that the program gives is a biologically relevant solution. Could be the second, could be the 10th, could be 1,000 later. And you see that after, if you want to generate 10 solutions, then the MILP implementation really struggles. So here, looking at the results, indeed, there are a few timeouts for the MILP implementation of Cobra Pi to get 10 solutions. And our tool is the only one that satisfies both network expansion and flux balance analysis constraints. So that is actually very useful for non-model organisms when you really want to study, to explore the solution space. Going back to our communities, not only working with a single population, but with multiple populations, we want to know what may occur in bacterial consortiums, in microbiomes. So that requires screening the metabolism in large communities. So as a reminder, from the genome, we will build metabolic networks having a few thousands of metabolic reactions. That's what we call genome scale metabolic networks. And when we look at the bacterial community, that's the first thing that we will do, build the metabolic networks for each of the genomes. Ideally, we have the information of available compounds in the environment, so we can predict what is reached in each metabolic network. And in particular, when there are several of them, we want to assess the complementarity of the networks, whether what is produced in one could be useful for the second one. And then if there is a function of interest that we want to study more deeply, we would like to identify the species that are associated to the production of these compounds. So here, the G compound, it is produced by the blue bacterium. Obviously, it's in this network, but without the compound D produced by the green bacterium, there is no way that in this environment, the blue bacterium produces the G compound alone. That is such things that we want to identify. So we built a pipeline called MetaG to Metabo that is dedicated to perform screening of metabolic functions in microbial communities. It starts from a description of the microbiome as a collection of genomes. So let's say you have several samples, you will have several microbial compositions, so several runs of this tool. Metabolic networks are automatically reconstructed. Then we predict the metabolic capabilities of the community and of each member. We identify which compounds get producible through cross-feeding, cooperation, interactions. And then we can predict minimal communities associated to certain functions. So overall, we systematically screen the metabolic potential of the community. So just to give you a bit more details in an example, here you have 6 metabolic networks. So the compounds are the circles, and the reactions are in the arrows. Each of the each of the metabolic networks has colored circles. So the yellow ones are the descriptions of the seeds, so what is available from the medium, and the green compounds are the compounds that we are interested in, that biologists want to understand the production of. So what the tool can do is first to describe what is producible by each member individually, given the environment. So here it's a toy, so it makes no sense biologically, but you have two bacteria in this case that cannot produce anything by themselves. And what we are interested in is the effect of cooperation, of cross-feeding among the community members. And because a species can produce compounds that will be used by others, it will unblock metabolic pathways and lead more compounds to be produced. So now more compounds can be reached by each of the community members. From this, we can select minimal communities associated to a few compounds production. Here, the green compounds. The tool will tell us that you don't need the 6 bacterial species to produce the 2 compounds. Actually, 3 is enough. 3 is also the minimal size of the community to produce them. And it can give you a solution. So here, a solution has bacterium 1, 5, and 3. We have tools that will highlight which interactions would be needed to sustain the producibility of these compounds. That would be the purple arrows. So exchanges of metabolites across species. But again, a first solution provided by a tool is not necessarily a solution that makes sense biologically. So the interest of reasoning approaches of the solvers that we use is that they are very efficient at exploring the solution space and enumerating solutions. So here we can very easily get the information that there are 4 equivalent minimal communities of size 3, that they have varying number of metabolic interactions, but that is very easily obtained. So here are our 4 minimal communities. When we look at them, we can see directly that there is one member, bacterium 1, that is present in every minimal community. So that is already very important information. Apparently, it has some functions that are necessary to produce the compounds of interest. And then the others, there are what we call alternative symbionts. There are in some minimal communities, but not all. We would like to understand how these symbionts, these community members may interact in the minimal communities. So the simplest thing that we can do is building association graphs, like the one on the bottom left, in which we connect 2 community members if they occur together in one of the minimal communities. But this generates very complex graphs at scale because there might be hundreds, thousands, or even millions of equivalent minimal communities because there is a very high functional redundancy in the microbiome. So we compress these graphs in order to have a better readability of the association of symbionts and to identify groups of equivalent microbial populations in the minimal communities. So here it tells you that each of the 4 minimal communities has always symbiont bacterium 1, either 2 or 3, and either 5 or 6. So we know which group have equivalent functions. And we actually tested it on real data, and we identified interesting patterns of members having similar functions with respect to the biological objectives that we targeted. So to finish, I will present you an application of the last tool to data retrieved from the Atacama Desert. That's a work that was done in the context of the Inria associated team, Symbiodiversity, that associates 2 Inria teams in France, so Dilis in Rennes, Pléiade in Bordeaux. And we work with the team of Alejandro Mas in the CMM in Universidad de Chile, and we are also associated to Inria Chile here. So in this project, we studied soil microbial communities in the Talabre-Leia transect in the Atacama. We had samples at 6 sites with physicochemical measurements for each, And we had shotgun metagenomic data for each sample. So this is the work of Constanza Andreani, who is now doing a PhD in France. So here is the data. So just a reminder on metagenomics. We extract DNA, we sequence it, either with short or long reads. Here it's short reads. We assemble the data. Then we can then reconstruct genomes, and from the genomes, we can build metabolic networks. And here we are interested in two types of data. The metagenome, that is directly the result of the assembly. It has some more or less large chunks of DNA, some that are very small. And we are also interested in metagenome assembled genomes, or MAGs. So that is roughly the genomes that we were able to reconstruct from the data. So from the metagenome, we have no idea which species are carrying the genes, the functions, but we have most of the functions. In the MAGs, we have information on the taxonomy of the species. So we can associate taxa to functions, but we lost all these small chunks of DNA that actually carry some important information. So we designed a systems biology framework to explore the metabolic potential in this Atacama transect by looking at both the metagenome level and the genomic MAG level. So we built two systems. We designed 5 conditions in order to test the effect of the sparsity of nutrients in the models. So we had very poor medium and very rich media conditions. And we performed simulations in order to get the information on reachable compounds at the metagenome level or at the MAG level for each of the conditions. So the first observation that we get, and that is expected, is that much more molecules are expected to be reached in the metagenome compared to the MAGs, which means that we lose a lot of information when we consider only the genomes. So we also see differences among conditions, simulated conditions. There are some that have a lot of compounds and unable to reach much more compounds to have a larger metabolic potential. Then we looked at the content of these producible molecules. So on the left, you have the result at the metagenome level. And you can see, so on this plot, the condition is the shape of the point, and the site of the transect is the color. And you can see here a clustering by shape and not by color, meaning that at the metagenome level, the effect of conditions is greater than the effect of the site. In other words, the sites cannot be very well distinguished at the level of the metagenome across functions. It means that there is a shared reservoir of functions in each of the sites. And this is not what we get when we look at the genome level, so the MAGs. The MAGs, they represent prevalent populations that were abundant enough so that we could reconstruct that genome during the assembly process. And here, the content of producible metabolites would cluster by color and not by condition. So they would cluster by site, meaning that the most prevalent populations retrieved in each site, they actually have different functions. Their functions differ across sites. So that is interesting because if the reservoir is very similar, actually, because the environmental conditions vary a lot across the transects, what we observe is that the most prevalent functions are actually very different across sites. So I will conclude on what we do in metabolic modeling in the team. We really have different approaches depending on whether we are working with large-scale communities or small controlled communities. In the first case, we need to generate models automatically. So our strategy is to use discrete models in order to reduce the complexity of the data, to make some predictions from which we can maybe perform more manual curations, and we can bring more expertise. So we decrease the complexity in order to target some functions, target some community members. And then for small or controlled communities, we use numerical models because we can curate the models. We have more expertise, we have more literature. We perform dynamic simulations, and we are not yet up to the digital twin, but this is the path that we are going forward. So overall, you've seen that we use a wide range of methods, ranging from machine reasoning to numerical models or statistical learning. And really, again, the choice of the model depends on the question and the constraints we have to address. So I want to thank all the people who were involved in the works that I presented. So on the left, you have the members of our team in Bordeaux, Pléiade, Simon Labarthe, a researcher, Coralie Muller, who was an engineer and now is doing her PhD thesis with us, Maxime Lecomte, a former PhD student, Shamnam, current PhD student, Pablo, former postdoc. We are an Inria and Inrae associated team. That's the particularity of our team. At the CMM, the work was mostly carried by Constanza Andreani with the help of Natalia Jimenez and Ricardo Palma and other colleagues, among which Alejandro Mas. The Entero Signature project was carried during my postdoc at the Quadram and Erlam Institutes in Norwich, UK. And I was in a team led by Farkil de Brand. And also other French collaborators, Anne Siegel, who is here, Arnaud Belcourt, who's now a postdoc at Inria Grenoble, Hélène Valentin from Inrae, and Loïc Polvé from CNRS in Bordeaux. And thank you for your attention.
Yeah. So I was wondering, on the kind of application or potential contributions of machine learning to your work or the works that you presented. So, for instance, in the large scale part of the work, so how I'm assuming that for instance, in the reasoning part, in the logical way of doing it, there's a lot of kind of limitations for doing that. In terms of beforehand, I assume that you need to write a lot of like restriction in terms of the biological processes. So what is your view in terms of applying, for instance, active learning or reinforcement learning for doing some kind of that work in terms of act as a filter or help you to look at what kind of be like candidate processes, candidate solutions. Regarding the low scale data or processes, how you try other type of like approaches like graph neural networks for modeling the data.
We haven't tried other models. Questions? You're right that there are some constraints before we use the reasoning models, but in fact, the difficulty really lies in describing the mechanisms in terms of logical rules. So transforming what we know about the biology into this paradigm that we use. So for instance, how can we describe metabolic cooperation, cross-feeding, the fact that one species will produce something that will be used by another species? Or can we describe competition for limited nutrients? So I think that is the most difficult part, because preparing the data, there has been a lot of development over the past 15 years to produce metabolic network, to generate them more or less automatically. So there is a wide area of tools that we can use from the genome to the metabolic network. So really, I think the development of the model is more complicated, and producing software programs out of it, because that's the part of the expert, and the users, they will not have to design these models. They will hopefully be okay with the constraints that we developed, and they will directly give the metabolic networks or just the genome. So does it answer your question? And then related to decreasing the complexity, that's really the purpose, yes. Because we hide a lot of complexity in our discrete models. We do not take into account the stoichiometry of reactions. It's just there is some A in the reaction. There is not one molecule or two molecules of A. We don't know how much. We make simplifications. Actually, when we model cooperation, we do not take into account transport reactions because they are not very easily annotated in the networks. So we make the assumption that one compound will be available for another species, but then it has to be checked afterwards. So we generate some models, and the idea is to restrict their numbers using biological expertise, using literature, in order to get only those that are compliant with what we know about the system.
I'm interested. I don't know if you were in the initial in the origin of this investigation project of metabolism modeling, but I'm curious of what is the motive of this project or this line of investigation. What was the motive behind starting this investigation? I was while I was hearing, I noticed that this is important to find microbial, ¿cómo se dice? Microorganisms of that produce compounds of interest, but I think I can visualize like what are important applications of understanding this metabolism of this community of microorganisms. Like I'm interested in I don't know if you know what's the like maybe the main purpose of this investigation.
One answer is related to the framework of systems biology. So I told you that you can study an organism through its individual components. So I could decide to study only the genome of the species, or just the metabolome, the molecules that are produced by the species, or the proteins, each component. And if you do that, even if you do all of them, you will miss the interactions among them. And that is important to understand the systems by itself. In systems biology, we try to integrate all of these components, and it turns out that the metabolism is rather a good level to integrate much data. So here I've mentioned mostly genomic data, but actually you can refine metabolic models with additional data. So you build them using the genome. You can constrain them using metatranscriptomic data, so the information related to the genes that are expressed. You can also add further constraints using proteomic data or metabolomic data, that is the compounds that were identified in your system. So it really is a good level to integrate much data in the context of systems biology. So even if in practice, we cannot always do that, especially when we work with large-scale systems, when you do this for well-known species, like it has been done for E. coli, for instance, you can really understand all of the individual components through the lens of metabolic modeling.
Thank you, Clemence, for your talk. I have 3 questions. In the first slide, you showed that for instance, some drugs or diets were correlated to some serum, like firmicutes or other. And this model can give you like a perspective about what pathways are affected. For instance, if diet is positively correlated to firmicutes, is it because it's affectingly to firmicutes or is affecting negatively to the other ones? And the second question was, if I understood well, in the middle of your presentation, you told that some environmental parameters were useful to predict fluxes, right? So how many environmental parameters you used and you could predict the whole system, the whole all the fluxes? And the last one is regarding to, in all this model, how do you deal with these genes, especially with the metagenomic data, all these genes that you with unknown functions? Thank you.
One regarding the entero signatures. Yes, indeed, we could associate the importance of the relative abundance of some entero signatures to some metadata of the host, and especially firmicutes was associated to healthy phenotypes. So it really is the prevalence of firmicutes that is associated to the good phenotypes. So clearly it comes from this group. But then for the metabolic pathways, it's not something that we've been studying in depth in this project. We started a bit to build metabolic networks for some members that we reconstruct the genomes and that were associated to the signatures. So we could see some interesting information, for instance, that some entero signatures were more complementary at the metabolic level to other signatures. But we haven't gone in much details. And I think it's really interesting to be able to characterize more deeply the metabolic pathways of each member of the entero signatures. Then the questions related to fluxes. We can predict the activity of metabolic reactions, so that those metabolic fluxes, there are concentrations of by a unit of time, by unit of dry weight, bacterial mass. And these predictions, they are made from a description of the environment. So that is to say, there are some metabolic reactions connecting the system, the cell, with its environment. They are called exchange reactions, and they represent the available nutrients in the system. So for instance, we will constrain those reactions in order to represent the fact that some nutrients are more or less available. And this is what we use to predict in the end some values. And most of the time, the values that we predict is the biomass of each community member. So the population growth of each member. And that is really what we can compare to available data. So for instance, we had bacterial numbering in the cheese project. We knew exactly at each time point the quantity of each bacterium. And that is what we can compare to the predictions of our model. Does it answer to the second question? Yeah. And the third question. Yes. So that is really a huge problem that I didn't mention. Here, we build the metabolic networks from the data that is available in databases. So I told you that there are many tools that enable the reconstruction of metabolic networks. Each of the tools is associated to a metabolic database. So you have Metacyc, you have BIG, you have ModelSeed, many of them. And these databases, they represent what we know right now related to the metabolism, what the literature contains in terms of description of metabolic reactions. And indeed, many metabolic genes, we don't know what they relate to, at least 30% of them. So this information is not present in the databases. It won't be present in the metabolic networks, which means that the predictions that we do are incomplete, and they are only complete with regards to the state of knowledge that is available in the database currently. So I really think that there is a long road ahead in order to identify those missing reactions about which we don't know much yet. And this is especially true when you work with ecosystems that are not much studied, like the ocean, the soil microbiomes. Many of the members in these communities, they hold metabolic reactions or metabolic genes about which we have no information. And it's a shame, actually, because the interesting functions, there are among these genes probably. We often speak about primary metabolism that is designed that is representing the survival associated function of the species. And then secondary metabolism that encompasses functions not necessarily associated to survival, but to other processes. And this is among secondary metabolic pathways that we retrieve interesting antimicrobial peptides, future antibiotics, compounds of interest for biotechnology. So there is really a need to better characterize this kind of metabolism, and it will be addressed at a certain point by computational models, because we can connect some genome mining databases to the metabolic network. We can better connect metabolomic data. That's actually the topic of Coralie Müller's PhD. But then a lot of work is also needed experimentally to better understand these functions.
Hi, thanks for the talk. I was thinking about this generation of minimal communities, especially with cross-fitting. And I was wondering if with these graphs of minimal communities, one could build some sort of fitness for the bacteria. I was thinking about if you can apply adaptive dynamics to these systems of bacteria to choose or know more information of how is the interaction between bacteria, for example, with these alternative bacteria in these minimal communities, that if they are competing or they are in a cooperative way, but in other community. So I was wondering if one could build some sort of kernel of interactions with these bacteria in terms of fitness.
And in fact, yes, the purpose of our operation is, well, reduce the complexity, get some hints into which members are interesting, which members we should study in more depth. So the idea is to look first at the essential symbionts, because of course they are needed for the dedicated functions that we study, and then combine them with others depending on, I don't know, the availability of the species in the freezer for experimentation, or the constraints of biologists. Maybe in one of the equivalent groups, the biologist will eliminate some members because they make no sense biologically, or because they are very difficult to grow. And then based on this, we can build more precise models, maybe numerical models, dynamic models, in order to make some more precise predictions before going to the experimentation. And then regarding your suggestion, we've actually had a project during Shabnam's internship a few years ago, trying to understand the mechanisms that are associated to a given equivalent group. If several members, several species are grouped together, it's because they provide to the minimal community a similar function. It can be the production of a compound, it can be the production of a precursor that is used by another member of the system. And the approach that we took is to use reasoning again in order to discriminate the functions that are present in one group and absent in the others, in order to target some mechanisms that could explain the association of the members. But we've not yet finished the project, and I think we could go much deeper to understand which interactions occur within a group or among groups.
Well, we actually went over time. Thank you very much. We don't have. I have some other questions that maybe we can talk about it later. Thank you very much, Clemence, and well, let's see if we have opportunities to keep discussing this. And there is, I think, an overlap of on the stuff we are doing. You see the reaction of the team and the topics you are working here also with Ansija. Thanks.